Okay, so we're going to talk about digesting and absorbing nutrients in this lecture. We're going to talk about the nutrients listed here on this slide. So carbohydrates, proteins, lipids, vitamins, minerals, and water. So beginning with carbs. We eat a lot of carbs, right, unless you're on a special diet. So most of our carbs are going to be consumed as disaccharides or polysaccharides. I've listed for you in the notes that the disaccharides are sucrose, lactose, maltose. The polysaccharides listed, starch and glycogen. And then other polysaccharides from plant material such as cellulose aren't digested. So we can digest sucrose, lactose, maltose, starch, and glycogen. What that means is we have the enzymes in our body creates these enzymes to digest these molecules. We can cleave bonds between the disaccharide monomers and create monomers, right? Because monosaccharides are the unit that we need to absorb through the intestinal lining wall into the bloodstream. Cellulose is a polysaccharide that we cannot digest because we don't have the enzyme for it. So, all humans, we don't create cellulase, which is the enzyme that breaks down cellulose. And so when we eat it, it just remains intact and it goes through our digestive system, does not deliver any calories to us. We don't absorb it. And so we call that fiber. All right. So plant fiber is mainly cellulose that you're eating in your diet. And there's no calories because we can't break it down. So let's start talking about how we do break things down, beginning with saliva, so at our mouth. So in our saliva, we have salivary amylase. Amylase digests starch, and we have amylase two places in our body. So we have salivary amylase, and we have pancreatic amylase. So enzymes are proteins. Amylase, when you swallow amylase from your mouth, it's going to get destroyed in your stomach. So it's no longer going to be active once it gets to your stomach. So there's a second amylase present, and that's going to be the pancreas, right? The pancreatic amylase. So what amylase is restricted to do is it can break down starches, right? But it can't break down starches from the ends. It's limited that way. And it can't break down starches when they branch. So what happens is the amylase will start to cleave these bonds. But at the branch points, what you're going to do is create these like short little, I guess they're called oligomers, but these short units of just a few glucose molecules, and those are called limit dextrins. And then since they can't chew on the ends, then once it hits the end, it's going to leave two glucose monomers together, two glucose monosaccharides or two glucose molecules, and that's called maltose. Okay, so when you digest starch with amylase, you're always going to end up with limit dextrins and maltose. And then further on down the line, your small intestine will start to break those down. So if you just want to skip down in your notes where you'll see the small intestine letter C, you can see that we have dextrinase, which is going to break down our limit dextrins. So this is dextrinase. And this is secreted by our small intestines. The brush border of it. So remember that the brush border are those the microvilli on the surface of those epithelial cells, and that's to increase surface area for pulling in all their nutrients. So those microvilli will have this enzyme to break down these limit dextrins. And then your maltose is broken down by maltase, right? Again, on your brush border. So other things that are in your saliva before we move on are lingual lipase, which is going to break down fats. So right away in your mouth, you're breaking down two of our most calorie or caloric foods. We have starches and fats being broken down because we want to extract as many calories as we can from our foods. We also create lysozyme. And lysozyme is an enzyme that actually destroys a molecule called peptidoglycan. So in this picture, this structure here, right, the orange and purple with yellow, this is peptidoglycan. Some people say peptidoglycan. But the lysozyme, we make this in our body. So it's really cool, I think, that we have evolved the ability to make an enzyme. So in our DNA, right, there's this instruction to make an enzyme that specifically breaks down bacteria. So we've, coexisted with bacteria from day one. We've evolved the mechanism to kill them with an enzyme that is secreted all through our body, really, in the largest amounts in our saliva and also in our tears. But it is secreted throughout our bodily secretions, so it makes sense that we would want to have some kind of antimicrobial molecule because, when we take things in from the outside, when parts of our bodies that are exposed to the outside, like our eyes, will have some kind of protection there. So that's lysozyme. So you don't need to know any detail from this picture, but this image is a bacterial cell wall. And when lysozyme is used to rupture the cell wall, the bacteria are easily lysed. So they break open from their osmotic pressure. They just kind of blow up. A lot of substances in your saliva. I listed a few: immunoglobulins, nitrogenous products, urea, ammonia. Bicarbonates are in there to modulate the pH. Acid is always bad for your teeth, right? Acid's bad for your teeth because it erodes the enamel. So when you eat acidic foods or if you eat candy, the microbes in your mouth actually break down that candy. They ferment the candy, the sugars, and then they produce lactic acid, and that lactic acid erodes your enamel. So when you eat sugar and candy, it indirectly erodes your enamel because the bacteria in your mouth will create acids. Anyhow, lots of things in there. Minerals are in your saliva. So when you do have that bacterial biofilm and you don't, floss regularly or every day, you can get that tartar buildup in between your teeth, around the gums, and that's the hardened biofilm that's created by bacteria. All right, so the small intestine, I have listed the enzymes that you should be familiar with, and then carbohydrate absorption. So let's take a look at this. Glucose and galactose. Glucose and galactose are the two monomers when you break down sugar. So sucrose, this is table sugar. And those two monomers are absorbed through the apical membrane. So again, here's our brush border. This is our microvilli. And it's microvilli. It's not cilia, okay? So here's maltose. Maltose is two sugar molecules, two glucose molecules. And so maltase, right? I told you how the enzymes are actually on the brush border. So those will transport them in. The monomers will come into the cell, and then the monomers will be transported across the cell to the bloodstream on the other side. So this is transcytosis. Well, it's just moving all the way through the cell. And it's active transport, secondary active transport, because we can see in this picture as well, we're creating our potassium and sodium concentration gradients with our sodium-potassium pump. And we can see that when we take glucose in, it's also traveling in with the sodium. So this is going to be secondary active co-transport, right? We're moving them together. And then just a quick thing on fructose. The sugar in fruit is absorbed through facilitated diffusion. So there's no picture on that, but it just comes into the cell. And then we have lactose intolerance, just a word on lactose intolerance. So lactose is a disaccharide, and it is made of glucose and galactose. I think I said sucrose was glucose and galactose, but sucrose is another two. Anyways, sorry about that. So lactose is going to come in, and it's going to get broken down by lactase. And those two monomers can get absorbed by your intestinal wall and go into the bloodstream. If you're lactose intolerant, the lactase is not present. So you have the genes to make this enzyme, but for some reason it's not being expressed. It just stopped getting made. So the enzyme is not present on the walls of your small intestine. So the lactose comes through your small intestine, enters into the large intestine, and then that is where you have a whole lot of what we call the microbiome, or our normal flora, the bacteria that live as residents in our gut, in our large intestine, in our colon. Normally, they really don't. They do a lot of good there, but If you're lactose intolerant, what happens is lactose comes into the large intestine. It feeds the bacteria that are present. The bacteria love lactose, especially E. coli, which is in large amounts in your colon. Coli is really for colon, so they discovered this bacteria and they named it coli because it was in your colon. So it ferments it, and then when the bacteria ferments the lactose, it creates acids and gases. And so the flatulence that people feel when they're lactose intolerant, this is from the gas. They can have abdominal pain from the distension of the colon from that gas, right? You're kind of pushing against the wall and that can hurt. And then also, what's not pictured here, but you have diarrhea. And this is because lactose is actually going to contribute to the osmotic gradient because it's a solute. So if you have a lot of lactose inside the lumen of the intestines, remember that your large intestine, the primary role of the large intestine is to reabsorb water. So it wants to take water from the lumen and wants to put it back into your bloodstream. But the presence of lactose disrupts that osmotic gradient, so you actually don't lose that much water, or you don't reabsorb that much water, right? So the process of going back is going to be smaller, and you'll get a lot of water. You'll have watery stool, which is basically diarrhea, right? So that gives you the symptoms of lactose intolerance. The acids and gases, the gas primarily will give you flatulence and the pain that people feel from the bloating. And then the lactose being present disrupts the osmosis process, and water is not going to get reabsorbed as much as it should. So you'll have more water in your stool, and that is diarrhea. So all because of that little molecule lactose. All right, moving into proteins. So proteins are going to be mainly from our diet, right? So when we take in foods, we eat protein-rich foods, as you can see here. But there's other sources of proteins that you may absorb through your GI tract, which is the enzymes that are secreted from your lining of the intestines or from your stomach. Those are proteins, and they can be broken down and then taken back into your intestines, right, into your bloodstream, broken down. Or they can be, which is kind of gross, but the cells that are constantly coming off of your intestinal, the whole GI tract, because remember, this is all epithelial cells. They are constantly reproducing, and they're going to get sloughed off. And so you can actually use those proteins and reabsorb those. So 3 sources of the proteins that you can actually get. How do you digest proteins? We digest it with enzymes as well. So there's 3 listed. Endopeptidases. Endo means within, so they're going to split that long amino acid chain in the middle. Exopeptidases will come at the ends, so they're going to cleave them bit by bit from the ends. So if I drew a line here, so endopeptidases would cleave in the middle. The exopeptidases will start chewing the amino acids off on the ends. And then there's a third class called zymogens, and zymogens are inactive forms of enzymes that need to be activated by another molecule or another substance. And the pancreas secretes all zymogens. So I gave an example of a couple. Pepsinogen is the inactive form of pepsin. And this is the picture of pepsinogen. So you can see that we have these chief cells. This is a picture of your stomach, the gastric mucosa. Chief cells will make pepsinogen. So that's a zymogen. It's an inactive form of this enzyme. And it takes hydrochloric acid to modify, to take off one of the pieces of the pepsinogen to make it active. So it's going to become pepsin. So pepsin is the active form. You can see pepsin is going to work and starting to break down the proteins. Hydrochloric acid is made by a different cell in your gastric glands, and those are called parietal cells. Okay, so chief cells make pepsinogen, parietal cells make HCl, hydrochloric acid. Let's take a look at the pancreas. So the pancreas is going to make 3 different kinds of zymogens. In your outline, letter C, trypsinogen, which you've probably heard of from anatomy, chymotrypsinogen, and then procarboxypeptidase. Very difficult to say. But what happens is, so here's the pancreatic duct. Here's the lumen of our small intestine. If you remember where this is the duodenum. So here comes the zymogens, right? The inactive enzymes. And trypsinogen is going to get converted to the active form, trypsin, by an enterokinase that is physically part of and attached to the wall of your small intestine here. Okay, so if you look at the brush border proteases, enterokinase is bolded for you. It's on the wall of your intestines and it converts trypsinogen to trypsin. Trypsin then goes ahead and activates the other two zymogens. Okay, so trypsin comes in, converts chymotrypsinogen to chymotrypsin, and it also converts your pro-carboxypeptidase to carboxypeptidase, and then those are active and they can start digesting the proteins. Okay, so what's key here is this enterokinase to start the whole cascade of events. How we absorb, I don't have a picture of that, but how we absorb amino acids is by secondary active transport or facilitated diffusion. And what's coming into the cell is either di or tripeptides, so very small units, right? So a dipeptide, here's a dipeptide, or a tripeptide. Those are what comes into the cells. All right, so we'll look at lipids. So lipids are interesting. Lipids are going to be a little bit more complicated because these are going to be fat soluble, right? They're not going to just readily dissolve in the water. So there's a special transport system for them. You may remember this from chapter 21. So most of the fats, or I should say lipids, most of the lipids that we take in every day is in the form of fats. But then there's also cholesterol. So you will be taking in some cholesterol, depending on your diet, of course. But the lipids, right, so I have that they're not water soluble, they do not mix with the stomach, they do not mix with intestinal contents, and they tend to form fat droplets, just like if you added oil to vinegar, right, they don't dissolve. So the trick would be to take those, they're going to naturally produce fat droplets. And so what you have here is an image of bile salt and fat. Let me see. So let's take a look at this one. So this is your fat globule, okay? And your lipases, the enzymes that are going to dissolve your fats. Remember, your fats are triglycerides, right? So the glycerol head, glycerol head, 3 fatty acid tails, right? This is a triglyceride. So remember, these enzymes, these lipases, can take off these fatty acids, and it can remove the glycerol, and it can create, these fatty acids, these monofatty acids. That's what lipases do. But lipases can only work and chew at the edges of this fat droplet. So they're going to be sort of limited because all the fat on the interior is going to be protected, right? So what you want to do is you want to break up those fat droplets, and that's what bile salts are for. So bile salts, these are amphipathic molecules, meaning that they are going to have a fatty part. They're going to have a hydrophobic part, and then they're going to have a hydrophilic part, kind of like a phospholipid. And they're going to be able to insert themselves in the fat. So the hydrophobic part will insert themselves in the fat. The hydrophilic part will associate with water, and it creates little tiny beads or droplets of the fat. So you go from one very large globule of fat. When you add bile salts, you're going to then break up this really large globule into smaller droplets. So globule to droplets. This is going to increase surface area for those lipases. So now the lipases have more surfaces to work on and break down those triglycerides, the fats. Okay. So the word for this is emulsify. Bile does not enzymatically break down the fat globule. It doesn't do anything to the actual triglyceride. That's the job of lipase. What bile does is it breaks down this really big globule with a limited surface area for those lipases. It breaks it into smaller droplets with a larger surface area for those lipases to act on. So that's called emulsification. When you emulsify a substance, you break down a really big droplet into a really big globule into small droplets. So yeah, bile emulsifies fats. All right, so let's take a look at this slide. This is your book's image of a bile salt. It has a hydrophobic component to it and a hydrophilic component to it, which is in blue, telling you, hey, this dissolves in water, this dissolves in the fat, right? So when you see how it sort of bonds with the fat, the yellow side faces the fat and the white side faces the water. The blue side faces the water. All right, so when we take a look at this, what happens is you're going to take in these fats from your diet, right? This is our nice fat droplet now. We can see that we have bile salts surrounding our fat droplet, and then inside our fat droplet we have our triglycerides. Okay, so now lipase, right, from our pancreas is going to come and it's going to degrade those triglycerides. Those triglycerides are going to be broken down to monoglycerides, all right, that's long lines of fatty acid. And then the glycerol is also not shown here, but the glycerol can be digested. But the glycerol is remaining to some free fatty acids and monoglyceride has a glycerol on there. The free fatty acid does not. What your cells will do is they'll take up either free fatty acids or that monoglyceride, okay? And then other times the remaining will just form little bubbles called micelles, but these will eventually get broken down. So absorbing into the cells will be the monoglycerides and the free fatty acids. And what happens is they have to get remade inside the cell. So we had to break them down in the lumen so that they can cross the plasma membrane. They're small enough to cross. Then once they're inside, they go to the smooth ER and they're remade. So they're remade into triglycerides. Then you can't just pump a triglyceride back into the bloodstream because it's going to form those fatty droplets again. So what your body has evolved to do is to package them up into something called a chylomicron. The Golgi apparatus does this. So the Golgi apparatus will take your triglycerides, put them together with other lipids like cholesterol and proteins, and create a ball called a chylomicron that's able to dissolve in the plasma. So it's a ball of fat, but with enough protein on it to make it hydrophilic. So it's able to travel in your plasma. So here, right, we can see the chylomicron being through exocytosis, leaving the cell. And the reason why these chylomicrons don't go into the bloodstream, that does not happen because they're too big to pass through the capillary network. The capillary wall does not allow them in, but guess what does? The lymphatic vessels. So remember back to anatomy where you're learning about lymphatic vessels. Lymphatic capillaries are leakier, they're more permeable. They actually have these open flaps that allows larger things in, such as cells, bacterial cells. Cancer cells can travel through the lymphatic system more readily, but then chylomicrons enter into the lymphatic system. So here comes the chylomicron into your lacteal. Remember, the lacteal is that finger-like projection of the lymphatic vessel, lymphatic capillary, into each villus, right? Each villus of your small intestine contains a lacteal. So the nutrients, the chylomicron, comes into the lacteal and then it will enter the lymphatic system. The lymphatic system will eventually return this to the venous bloodstream, right? So it will go into general circulation. But what it does in the immediate sort of travels is that it does not go to the liver. Remember that all the other substances from the digestive tract, the blood, whatever goes into the bloodstream, will eventually go to the hepatic portal vein, right? And that portal vein will lead into the liver, and the liver can start to package up, detoxify, take off nutrients that the liver needs. And then that once it's cleaned and sort of processed, the liver has processed the blood coming from your digestive organs, it goes into your blood supply. But not so with this fat-soluble substances. So your fat-soluble substances like your vitamins A, D, E, K, fat-soluble toxins from the world, some pesticides and herbicides may be fat-soluble. They actually don't get cleaned by the liver first. So they just enter general circulation because they don't go through the bloodstream. They go through the lymphatic. They will eventually, of course, hit the liver. They will get, but initially, any fat-soluble substances go into the bloodstream. They don't go to the liver. They bypass the liver. All right, so a quick word on the different kinds of lipoproteins that we have traveling throughout the body. So lipoproteins are going to be a mix of lipids and proteins, again, so that they can travel in the bloodstream. And these are the different forms. So we talked about the chylomicron. So this is coming from the small intestine. The small intestine makes these. Okay. The others listed here are going to be formed by liver. So that the LDLs, they're going to be, the DLs, I guess. The lipoproteins, these other rest can be formed by the liver. So the larger they are, the less dense they are. So the chylomicron is the least dense form. And then the HDLs are smaller. They're the smallest kind of lipoprotein, but they're the most dense. So what that means in terms of density is that the more protein that's present, the ratio of protein to fat, the more protein that's present, you are denser and denser and denser. And the more fat that's present, you're less dense. So the chylomicron is full of fat, full of lipid, and it's giant. So it's the least dense and largest. Let's talk about the VLDL. So the liver, when it gets the lipids delivered by the chylomicron, it will form these very low density lipoproteins. This will exit into the bloodstream and then it'll deliver fat. So remember that on the surface of capillaries, especially rich near adipose tissue, but there was that lipoprotein lipase. Let me write down the word lipoprotein lipase. We talked about this in chapter 21. There's an enzyme that's on the capillary walls that will start to cleave the fats as the VLDLs come about. When they take the fat off, right, they enzymatically take the fat off so that the cells of your body can get fat, it will shrink and it will become an ILDL, intermediate density, and then that will run around your body, and that the lipoprotein lipases will start to then chip away and take fat off of that, and then that becomes even smaller, which is an LDL. So the low density lipoprotein. LDLs are known as the bad form of cholesterol. So it turns out that the LDLs will still go around your body. Now these LDLs, aren't not fatty anymore. They're actually more cholesterol because the lipoprotein lipases, these are taking off mainly triglycerides, okay, the fats. Your cells need fat. The cholesterol they stay inside the lipoproteins. So when you get down to the LDLs, this is mostly cholesterol now. Okay. When this is in your blood vessels, the larger blood vessels, it tends to deposit those cholesterol molecules underneath the lining. So here's that picture, right? Here's your cholesterol, LDL bad. And this is because the LDLs will actually put the cholesterol in this underneath the endothelium. And we'll start to build that cholesterol plaque, right? And that's unhealthy because it narrows the lumen, causes atherosclerosis, causes hardening of the arteries. Lots of bad things can come from that. HDL though, is the good form of cholesterol. So it can actually remove the cholesterol from that plaque. So as long as the plaque is not hardened, the HDLs can actually remove it. So you will always have a ratio of LDLs and HDLs in your bloodstream because this is the natural way that our bodies have evolved to carry cholesterol around. And so this is just a side-by-side comparison, right? What is LDL and what is HDL? So LDL can basically contribute to the formation of plaque. and that is going to give you a higher risk of heart attack and a higher risk of stroke. And so these are ways, right? You want your LDL be below 70 milligrams per deciliter. And then these are ways that you can help to lower your LDLs. HDL, high density lipoprotein, should be at this level 50. You don't have to memorize these levels, but some people can raise their HDLs by, again, exercise, quit smoking, avoiding saturated fats, and losing weight. Okay, and these are beneficial because, again, they can remove those cholesterol plaques. All right, let's talk about vitamins. So there are two kinds of vitamins, or two categories of vitamins. They are water soluble, so these dissolve in water, and then fat soluble, they dissolve in fat. So remember your ADEK, right? You want to remember ADEK is fat soluble. There are other names that they go by. So retinol, retinoic acid, these are all vitamin A. Vitamin D there's a form of vitamin D that's a really long molecular name. But anyways, it's cholecalciferol, but oftentimes you'll see like just D3. And then tocopherol, or tocopherol acetate, sometimes vitamin E. And then I can't even pronounce this, so vitamin K. What is happening when you eat a fat-soluble vitamin is that they get rolled into those chylomicrons. They get rolled into the micelles. And so your fat-soluble vitamins get absorbed with the fats. And they get delivered in the body, in those units. And so if you are wanting to have better absorption of your fat-soluble vitamins, you should have a little of fat with those foods. So, and you'll find that supplements will sometimes come packed with fat, like it'll be an oily kind of supplement because you need the oil, you need the fat in order to absorb it. For water-soluble vitamins, they're going to be actively transported. Your body wants them, they need them, so they're going to have all special transporters to pump them into the cell. I just want to point out intrinsic factor, which is not here. Or is it B12? Oh yeah, here it is, B12. So B12 is absorbed only if it's bound to intrinsic factor. It needs intrinsic factor to be absorbed. Absorbed. Okay, so and this is the intrinsic factor is produced by the stomach, and if you can't get the B12, B12 is required to make hemoglobin. So if you don't get enough B12, you're going to be anemic, okay, because it's required for hemoglobin. So low B12 will cause low hemoglobin, will cause anemia. And if it's because intrinsic factor is not present, then this is called pernicious anemia. Or it can be because your B12 is just low. So either way, you're either not eating foods high in B12 or your body's not making enough intrinsic factor. Either of those scenarios can cause pernicious anemia, which is different than dietary anemia, which is an iron deficiency. All right, let's talk about here's my picture on intrinsic factor. So there it is, pernicious anemia, lack of intrinsic factor, or B12. All right, and just a quick word. So B12 is in higher amounts in animal foods and animal products. There are fortified foods with B12. So if you are avoiding animal products, like you're a vegan or vegetarian, you need to be aware of this B12 issue. And look it up. There's foods that you can eat that are higher in B12 that are not animal product, but you have to be careful about that. Okay, a few minerals, and then we'll end part one. Calcium absorption. Actually, this is not the first one. So sodium, let's just go through the outline. Sodium and chloride, salt, right? Salt is passively absorbed through the small intestine. It's called solvent drag. Basically, when water gets reabsorbed, it also moves the salt. So you can see it's either passively absorbed with a leaky epithelium in the duodenum and jejunum, or it can be actively absorbed. So either way, we can passively or actively absorb salt. Potassium, not a whole lot on potassium. Just know that if you do have diarrhea, you can actually lose an excess amount of potassium, and then you can have a condition called hypokalemia. Bicarbonate is absorbed passively. If you have diarrhea, you can actually have a loss of too much bicarbonate, and that could give you acidosis. Calcium, so here we are on calcium. Actively absorbed in the duodenum and jejunum based on your body's needs. So the brush border here, right, will have a transporter, and you don't need to know the name, but there'll be a transporter for calcium. And then once that calcium is inside your cells, it needs to bind to a calcium binding protein called calbindin. You don't need to worry about DN. But calbindin binds to it, and it helps the calcium move through the cell, and then there's another facilitated transport out. Actually, there's a pump. This is an active transport out to the bloodstream. So this is how calcium moves through the cell. Okay, there's a transporter. There's a special binding protein for calcium to facilitate the passage to the pump, and then it gets pumped out to the bloodstream. Vitamin D, right, the active form of vitamin D, which is called calcitriol, or if you see that, this is what I was talking about, that large molecular formula, that's the activated form of vitamin D. So this guy here is also known as calcitriol. And it can, the presence of vitamin D3 is going to help calcium get absorbed because it has action on producing more calbindins, more transporters on either end. So it just facilitates the absorption of calcium. It enhances that absorption of calcium. So that's why we need D3. D3 is required for calcium absorption. In terms of iron, so iron is going to be bound to transferrin protein. So this picture, these are not in your textbook, but I just want something to look at while I was talking about iron. So iron is going to be iron, and then the transferrin protein. It's going to be taken into the cell through endocytosis. So you can see this is receptor-mediated endocytosis. So the iron comes into the cell, and then it can be released from the binding protein, okay? And then we are going to recycle, so we're following kind of clockwise here. Then we just recycle the transferrin and the receptor. And so we just have this receptor for iron. So we have a specific receptor to bring iron into our cells. Okay? So some iron can be stored as ferritin, and then other times, and then some iron can be moved out of the cell, bound to transferrin, that protein that helps move iron in the body. All right, water. So absorption of water is going to be primarily in the colon. The 7 liters that we have are absorbed by the small intestine, but the colon is going to really remove the 2 liters that, the 7 liters is reabsorbed, like as you're reabsorbing the nutrients. But then once all the nutrients have been absorbed and you're not pulling nutrients anymore, the colon is going to pull the water. So 2 liters of water absorbed by the colon. The water reabsorption is going to be passive, all right? So that's going to follow the solutes, and it's osmosis. That's why when I talked about lactose intolerance, how lactose can actually upset this balance, and the osmotic pressure is not going to be that great, and you'll have watery stool. What's really interesting here is that, just a picture to look at, your stomach is completely not permeable to water. So you can drink a hypotonic water or hypertonic solutions like Coke. So I have here, Coke is full of sugar and it's also full of salt. And so it's very, very hypertonic. So you would think that if this hypertonic fluid hits your stomach, that's going to suck out the water, right, from the lining of your stomach. But it does not happen because the stomach is completely impermeable to water. So you can drink any tonicity and it's fine. But your small intestine actually is not that way. So if you drink a very, very hypertonic solution like Coke, it can actually dehydrate yourselves through the intestines, but your stomach is not going to be dehydrated. All right, I think that's the end of part 1. Let's do some review questions. Where does the process of digestion by amylases begin? The answer is the mouth with saliva. Which of the following substances emulsifies fats? Answer is bile salts. Which of the following is not a lipid-soluble vitamin and therefore is more difficult to absorb, sometimes requiring active transport? Okay, so this is going to be letter B, B12. So A, D, E, and K are lipid-soluble. B12 is not. All right, so this is part 2.